In this video, we're going to take a look at the other exceptions to Mendelian genetics. So let's take a look at this pedigree here. So at this point, you have seen pedigrees before, and look at the patterns of inheritance. What seems a bit odd? So pause the video, think about it. And so if you thought about it for a bit, you might notice that whenever the mother has a disease, the kids tend to have it. So mother has it, kids have it. Mother has it, kids has it. Mother has it, kids have it. When the father has it, they don't pass it on. So there's something interesting going on here. And so this is another one of the Mendelian assumptions that we're going to break now. So Mendelian assumption is that genes are carried on nuclear chromosomes. In other words, chromosomes found in your nucleus. So they're called nuclear genes. And that is most of your genes, basically. They're in your nucleus. Okay. And so Mendel studied characteristics that were found on chromosomes found in the nucleus. But there are some genes that are called extranuclear genes that are not actually found in the nucleus. They're found in maybe the cytoplasm. Those are called cytoplasmic genes. And then sometimes other organelles like mitochondria and chloroplast in plants. So the extranuclear genes, they're actually found on those circular pieces of DNA called plasmids, which you've seen before. And their pattern of inheritance is different than you would expect for nuclear genes. Especially the mitochondria and chloroplasts, they're inherited by what's called maternal inheritance, which means that a zygote is going to get the extranuclear genes, especially those mitochondrial and chloroplast ones, from the egg's cytoplasm, not from the sperm. So that's why if the mother has a mitochondrial disease, like mitochondrial myopathy, where you get weakness, intolerance to exercise, and a lot of muscle deterioration, that mother will pass it on to the kids. But if the father has it, the father won't pass it on because the father does not pass on mitochondrial DNA to the kids, but the mother does. The egg has a lot more resources needed, so it has a lot more to it. So that's why it contains the mitochondrial DNA. Leber's hereditary optic neuropathy is a progressive disease that leads to progressive blindness in people as young as 20s and 30, in their 20s and 30s. That's caused by a mitochondrial disease, and that will be passed on by the mother to the kids. And there's evidence that mitochondrial diseases and mutations can actually lead to some cases of diabetes, Alzheimer's, and other heart disease. But again, that would all be passed on by the mother. So the main point here is that if Mendel had studied characters that were in mitochondrial DNA or chloroplast DNA, he might have seen different patterns, and that would have been a bit tricky to come up with conclusions with. So today, though, we know that there are extranuclear genes, and it's not just nuclear genes that exist, and that they do have their own patterns of inheritance. So here we can see a plant cell, and a plant cell has some mitochondria, so there's mitochondrial DNA, but also chloroplast. So here's a chloroplast, and inside that chloroplast, there's its own DNA on plasmids, and that's passed on in the female gametes when reproduction is happening. And so here you can see an example over here. This is actually caused by genes in the chloroplast, and it's inherited by the maternal parent, the female gamete in plants. So another assumption made is that there's normal meiosis. So basically, the characteristics will come out normal. There won't be any abnormalities to it. But as we know already, that there are oftentimes errors during meiosis, and those errors are called non-disjunction errors. So you can have non-disjunction errors that basically lead to abnormal gametes, and when the abnormal gametes are used to make an individual, you get what's called aneuploidy, where you have one missing or maybe one extra chromosome. If you look over here, we have a normal karyotype, but if you look over here on chromosome 21, we have an extra chromosome, chromosome 21. That's probably because we used a gamete that was made when there was an error in meiosis, maybe here or here, and that led to an extra chromosome, and it's called aneuploidy. So that leads to different phenotypes, and oftentimes in humans, it's developmental phenotypes, differences. So now, what's interesting, though, is that sometimes these errors of meiosis can happen, and they can lead to what's called polyploidy instead. Now, this is much more common in plants than in animals, but basically, if we look over here at this picture, this cell was 2n is equal to 6. There was a failure during meiosis, and now we have a tetraploid cell that formed. So 4n is equal to 12. When it makes gametes, the gametes will be 2n is equal to 6 again. And then when they combine, you'll have 4n. So what happens is this leads to the formation of an organism that was diploid before, but now that organism has become tetraploid, all because of an error in meiosis that led to basically this doubling of chromosomes, which leads to gametes that are 2n. And when you combine 2n together, you get 4n in the end. So you get an individual that is a tetraploid rather than a diploid. And so that's actually a method of speciation, if you remember back in evolution, where different species of plants can form when there's these errors in meiosis that create these polyploids. So again, it's more common in plants than in animals. You can see it in some animals. So this is a tetraploid animal over here, but not as common. So we can look at triploids. So bananas are triploids. They have 3 sets of chromosomes. Hexaploids, like wheat, 6 sets of chromosomes. And this is all because of errors in meiosis, which can lead to different species within the same environment. So sympatric speciation. So you can always review that and see that connection between genetics and evolution. This is a great video that goes over strawberries and their ploidy and why they're a certain ploidy, and it would be great to watch because you've done a DNA extraction. So in this video, we're going to finish off by taking a look at how gametes can be made. So if you look over here, we have a cell, we have big A, little a, big B, little b. And so they go through meiosis, and you get basically big A, big B, little b. And then sometimes the little b goes on the same side as the big A, and so you get big A, little b. And the big B goes on the same side as the little a, and you get little a, big B. So these are the different gametes that you can form. And that's how meiosis would happen. There could be crossing over and stuff like that, but we're looking at these 2 different genes. So we're looking at big A, big B, little b. Gene B, gene A, the alleles will separate, but they're independent of each other, these chromosomes. So big A can end up with little b, and little a can end up with big B. Now, that's what we see here. And so that's meiosis that we would typically assume. Now let's take a look over here. This is a bit different. Gene A and B were on different chromosomes here. But what if gene A and B were on the same chromosome? Because a chromosome actually has many genes on it. So what if they were linked by being on the same chromosome? Well, if that was the case, if they were linked, big A and big B would have to end up together. Little a and little b would have to end up together. So the gametes could not have this much variety, and so you'd only have big A, big B, little b gametes. Whereas over here you have a 25%, 25%, 25%, 25% chance of these different gametes. Here you only have 2 types. But then there's a small amount over here, 2% of big A with little b and little a with little b, even though they're linked on the same chromosome. How's that possible? Think about that. What happens during meiosis? If you said crossing over, that's right. So crossing over can happen, and you do have a small amount of the big A, little b, little b, but it's still an overwhelming majority of just this big A with big B because they're linked like that, and little a with little b because they're linked like that. So what does that bring us to? That brings us to the assumption that chromosomes assort, or genes assort, independently. So Mendel had studied genes for the most part that were on different chromosomes. And so it led to the patterns of, 3-to-1 ratio, 1-to-1, and if you've seen dihybrid, 9-3-3-1, which you'll see later, or you'll see, you've already seen. But the idea that genes are located on different chromosomes was what we were going for when we were talking about simple Mendelian genetics. In reality, genes can actually be linked. When genes are linked, what that means is that they're located on the same chromosome. And so their alleles don't assort independently because they're on the same chromosome. So big A and big B, they're not independent of each other because they're on the same chromosome. Whereas if we go back here, they're on different chromosomes. So big A could be with big B, but sometimes it could be with little b. They're independent of each other. So when they're linked, what happens is you get a little bit less of the other types of gametes, and you get just the ones that are linked. So you get the big A, big B, little a, little b gametes, and then just a small amount of the other types that could technically be made, all because of crossing over. But again, look, 2%, 2%, and this is 48%, 48%. So very, very small amount. The ones that are made just by separating, that's called parental gametes. And the ones that are made by crossing over is called recombinant gametes. But the ratios won't be as expected. So if you look over here, we have a situation where we have, if we had linked genes, or unlinked genes, sorry. So for example, if we had A and B on different chromosomes, in this type of cross here, you'd expect a 1 to 1 to 1 to 1 ratio. But when you have linked genes, the ratio is thrown off. Look, you get 7 to 1 to 1 to 7. You don't have to memorize that, but it's thrown off. And the reason it's thrown off is because you get a lot more of these kind of original parental gametes, big A, big B, little b, than you would these little recombinant gametes here. So you get a lot more of one type, these parental types sort of, made by these parental gametes than the other ones made by recombinant gametes, because crossing over doesn't happen too often to get the genes to switch. So that's another example of something that could have gotten in the way of the patterns that Mendel was studying. And we just learn more and more about linked genes, and we understand how they're inherited together, and percent crossing over as well. So your goal should be able to, at this point, go through this whole list here and basically explain what the assumption means and provide at least one example of the assumption being broken or violated by some characteristic or some concept that you know now. The only one you don't really have to do is the no imprinting. You can definitely research that on your own to see what that means, genetic imprinting, but really A to H is what you should focus on. So it's great to watch this video. This is in your notes. You can complete your notes and watch this video here and add some notes about the video. This goes over the characteristic of height and how it's controlled by several genes. Quite interesting. So please give it a watch as you do your learning goals and summarize your notes.